Good afternoon. I'm Marc Quinn, Hepatobiliary Research Fellow at the University of Liverpool, and I am delighted to present our AMMF funded study on the identification of actionable targets in peri-hilar cholangiocarcinoma using whole exome sequencing. So this work has now been completed and has formed the first part of my PhD thesis. So I'm going to talk a little bit about why we've focused specifically on peri-hilar disease, what we sought to achieve within the context of this study, the technology that we've deployed to assess this disease, the results that we now have, and the further study that is ongoing at Liverpool. So firstly, as a background, when we look through the literature on peri-hilar cholangiocarcinoma, or indeed cholangiocarcinoma in general, we can see that there has been that this disease has always been considered much less poorly understood comparative to the related HPV cancers, such as HCC and pancreatic cancer. However, over the past 5 to 10 years, there's been an explosion in next generation sequencing technologies analyzing this disease. But when we look deeper, what we see is that there is this huge disparity between intrahepatic disease and extrahepatic disease. So the overwhelming majority of work that has been done has been on intrahepatic disease, despite this being the least prevalent subtype globally. Now, global studies have been done, but again, the majority of work has taken place in the Far East, where we know we have differences in risk factor association. So in the Far East, we have parasite and hepatitic infections, whereas in the West, it's predominantly idiopathic. And this is important because this sequencing data ultimately translates into efficacious targets. And again, we've seen that in iCCA with FGFR2 fusions and IDH1 and 2 mutations. So what does all this mean? It means that we have this relative dearth of information on extrahepatic disease, and within that, specifically peri-hilar disease, and that what we do know has come mostly from the Far East. So thus there is this substantial ongoing unmet need for peri-hilar cholangiocarcinoma, and it is the most prevalent subtype. And what that ultimately equates to is that we have an absence of targeted therapies for it. So ultimately this posed the question as to what is the relevance of this to Western populations? And what is the relevance more generally to hilar cholangiocarcinoma? So thus we sought within our aims of this study to characterize the mutational landscape of a UK resected hilar cholangiocarcinoma cohort, to validate the presence or absence of Eastern mutations, to assess whether we could elicit actionable drug targets within these, identify any new cholangiocarcinoma mutations not traditionally associated with this disease, and take all of that data together and see if we could characterize the underlying biology driving cholangiocarcinogenesis in hilar disease. The technique that we employed is primarily whole exome sequencing, which is a next generation sequencing tool focused on DNA, and it looks at the protein coding regions of exons. And this is important because the exons we know contain 85% of disease-associated variants or mutations. And this is actually the first exome sequencing study that was done on a Western cholangiocarcinoma cohort. When we started, we had 45 formalin fixed archived samples, tumor and duct, and simultaneously we set up a fresh tissue acquisition protocol, which then boasted 20 cases by the end of my 2.5 years. All of these samples underwent consultant review and microdissection to boost tumor cellularity to ensure that what we were sequencing was indeed cancerous cells in the main. Following quality control, we had 25 samples that were able to go forward for whole exome sequencing, and 20 samples that were then subsequently underwent targeted sequencing, and that married up with the fresh frozen samples. Sequencing was done at the Liverpool Centre for Genomic Research, whereby the exome samples were sequenced on the Illumina HiSeq 4000 and the targeted samples on the ION PGM sequencing kit. We achieved greater than 50x coverage in the exome series and greater than 1000x in the targeted. And ultimately what it is that we're looking for within these data are those mutations that are significantly differentially expressed between tumor and normal tissue, the normal bile duct tissue. In terms of the bioinformatics, this data was then aligned to the human genome. When we talk about variants and mutations, we're using these terms interchangeably. But ultimately, what we're focused on is what's called variant allele frequency. So this is the observed sequencing reads over the total reads at that particular genomic locus. For somatic mutations, we considered those that were greater than or equal to 1% and significantly less than 50% to be considered somatic. Those that approached 50% or above were considered germline. We then took all of those variants and screened them initially against DB-SNP, which is the database of polymorphisms, and COSMIC, which is the catalogue of somatic mutations in cancer, which is administered by the Sanger Institute. Once we had a complete mutational data set, we then compared that with the OncoKB database for drug targets, which is administered by Memorial Sloan Kettering. But ultimately, at the end of analysis, we took all of the variant data that we had, coding and non-coding, and did a gene set enrichment analysis to see how all of these genes and their mutations interrelate together. So moving on to the results and focusing firstly on these sequencing statistics. We achieved on-target mapping in the exome series of 76% and in the targeted series of 99.5 to 100%. When we merged these data sets together, we then looked at what the median number of somatic mutations was per megabase, and that equated to 13.57 per megabase of DNA, with quite a substantial range. But notably, this did contain some outliers within that, notably there the 74.43. However, when we take all of this together, it equates to a moderate tumor mutational burden, TMB. And this is interesting because cholangiocarcinoma has historically been associated with a low tumor mutational burden in keeping with other HPV cancers like pancreatic cancer. And this is important because TMB acts as a surrogate marker for neoantigenicity and thereby tumor immune cell infiltration and ultimately immune checkpoint inhibition response. And we know from the Keynote trial that checkpoint inhibition responses have been limited in cholangiocarcinoma. But again, this is something that we will come back to a little bit later in terms of the ongoing work that we're doing in Liverpool. Moving on to the actual mutational landscape itself. When we compared what is contained in COSMIC, so a publicly available database, compared to what we found in Liverpool, we see some quite stark differences, particularly when we look at those mutations that are commonly seen in the cholangiocarcinoma setting. So TP53, very well characterized in most cancers. In COSMIC, it's present in 26.6% of cases, but that massively jumps to 52.4 in Liverpool. KRAS, fairly equivalent. But those other mutations that we're well versed in with cholangiocarcinoma, like IDH1 and 2, present in 10% of cases in COSMIC, absent at that 1% variant allele frequency in Liverpool. Same goes for BAP1. Another mutation that we all often hear about in CCA, ARID1A, present in 12% of cases, drops to 2.4% of cases. So this is showing us two things, that the landscape in Liverpool is differing from that what's gone before. But this, yes, we do have smaller numbers than what's in COSMIC. However, this also suggests that there is a substantial overrepresentation of intrahepatic disease within public databases, again, in keeping with what's in the literature. What gets more interesting is when we look at the mutations that are higher frequency in our series, particularly mTOR, ABL1, Notch1, PIK3CA, NF1, and EGFR. And each one of these is an established cancer-associated mutation, but is not really thought of as being a mutation associated with this disease. We're seeing them at quite significant prevalence. And we'll talk about each one of these in turn in just a moment. When we then took these mutations of interest and ran them through the OncoKB database, some of these have well-established therapeutics, FDA approved for those mutations. Others do not, but have evolving therapeutics at the time of this presentation. So this is interesting. So we have some agents that we will have heard of in a biliary tract cancer setting, but some that we will not have done. And we'll talk about these in turn. But what all of this data shows us is that there is an evolving hilar cholangiocarcinoma specific landscape. And we need to take that into consideration when we think of biomarker-driven trials, particularly within an adjuvant setting, as we're focused here on a resected cohort. So using mTOR as the first example, this is a pathway which has been seen in extrahepatic disease before, but specific mTOR mutations have been lacking. So we've seen mutations in genes upstream and downstream of this of mTOR, but not in this gene itself. So now we've got new alterations that we've seen in that gene. Now, many of you will have heard of everolimus before, having had activity in unselected biliary tract cancer. But again, do we need to be considering this in the context of stratifying hilar disease? And again, stratifying peri-hilar patients for those who have mTOR mutations. Neurofibroma 1, which is involved in regulating mTOR in the RAS-RAF pathways. Selumetinib is an agent which is FDA approved for neurofibromatosis, which inhibits the MEK enzyme. Has also had activity in unselected biliary tract cancer. Again, do we need to be thinking about stratifying patients with NF1 mutation within a hilar context? Some of the other mutations related to mTOR, like PI3KCA, is another mutation that has a huge amount of work ongoing in other cancers unrelated to biliary tract cancer, but are these other agents that we need to be considering in this context? And then mutations that we, for example, wouldn't have considered at all, such as ABL1, very well established in leukemias, but has also been shown to have increasing prevalence in solid cancers like non-small cell lung cancer. Now, afatinib didn't show any survival benefit in phase 1, but the prevalence of mutations in this gene in our series suggests that ongoing assessment of this type of drug, a tyrosine kinase inhibitor, is justified. Notch signaling, many of you will have heard of in the context of intrahepatic disease, overexpression being associated with that. In our series here, we've seen specific Notch1 mutations which haven't been identified in the literature before. Now, Notch can be oncogenic or suppressive. Its role is uncertain in this context. And again, this is work that we have ongoing in Liverpool. But this is important because this represents one of the agents or one of the gene mutations that has evolving agents at the current time. So there is a Notch inhibitor in trials in solid cancers at the moment. And this would suggest that a reasonably high number of hilar patients would be eligible for such a trial. So again, all in all, the message here is that there is a need for hilar cholangiocarcinoma specific biomarker driven trials, and an adjuvant context would be needed in addition. Exome sequencing as a tool is exploratory and capable of identifying new mutations of a set panel. And within our exome series, we identified a somatic mutation within a gene called mitogen activated protein kinase 9. Now, this gene, mutations in this gene are implicated in the development of esophageal and retroperitoneal neuroblastoma. And in the HPV context, interestingly, in pancreatic cancer models, inhibiting mutations within this gene inhibits the MEK and ERK pathways, and thereby reducing carcinogenesis in pancreatic cancer models. So what we have found is a CCT deletion. And we found that at a somatic allele frequency in 10 out of the 25 exome sequence tumors. And again, this was of low variance. However, we also found it at this one specific deletion at a germline frequency in 12 additional tumors in the exome series. So putting all of this together, this specific mutation may have driver status, and interestingly is targetable with a tyrosine kinase inhibitor, fostamatinib, which is traditionally used in the setting of thrombocytopenia. So really interesting. We have further validation work on this ongoing in Liverpool. And again, what this gene plot shows you is the huge amount of activity going on within this gene. So each one of these pie charts representing the number of patients with a variant at that specific locus. And this was certainly the most interesting of the genes identified within the exome series. So genes not traditionally associated with this disease. And indeed, when we look back through COSMIC, if you look for mutations within MAP3K9, We found it in only one patient out of a series of 400 patients. So again, cosmic databases focused on intrahepatic disease. We've got a mutation here which may be associated with cholangiocarcinogenesis in Hilar disease. When we took all of the variant data together, we did a gene set enrichment analysis. And what this has shown us is how all of these genes and the variants within them relate together. And we looked primarily at those genes which were statistically significant, i.e. with a false discovery rate of less than 5%. And virtually all of the genes above this level pertained to the host immune response within the tumor tissue and pertained to both innate and adaptive immune responses. The most enriched pathway overall was the dectin-2 family. And this is interesting because dectin-2 is expressed mostly on myeloid cells and has been implicated in the suppression of liver metastasis formation. And this is of potential critical importance in cholangiocarcinoma, given the often advanced presentation of the disease. So this innate response certainly warrants further investigation. Now, what you can see here in this core diagram is the edges connecting the same variant-enriched gene through multiple pathways. And we can see here at the top of that diagram that there is really quite substantial overlap between PD-1, CD3 phosphorylation, and the ZAP70 translocation pathways. And this is interesting because these are all T-cell receptor pathways. And what this network diagram here is showing you is the genes that are shared by all. And these all pertain, are the genes that are shared between all of them pertain to the major histocompatibility complexes, class 2. And what this is showing is, when we put it all together, are that there are new avenues here with these genes to functionally assess how host T-cell infiltration is impacted by alterations within these genes. And we're going to use this to explore how their up and down regulation impacts on T-cell infiltration, and thereby explore how this impacts immune checkpoint inhibition. So new avenues have been provided by this gene set enrichment analysis. This is a heat map, which again is showing the overlapping genes between these various different pathways. And again, we're seeing this very strong relationship as regards T-cell receptor activation pathways. So each of these genes we'll be focusing on in more depth over the coming months and years indeed. When we then looked at the cellular proliferation pathways within the statistically significant pathways, even these exhibit a host immune response role. So one of the most significant that we found was the neural cell adhesion molecule, gene 1, which actually regulates neurogenesis, but also has a role in immune surveillance via T-cell and B-cell expansion. Now, in the Cancer Genome Atlas, they found that polybromo 1 may regulate stromal cell infiltration via NCAM1. And this is very interesting, given the prevalence of polybromo 1 mutation in our series. So all in all, this study has shown that there is an absolute need for biomarker-driven trials that are specific to peri-hilar cholangiocarcinoma. This is an ongoing unmet need. What our data has shown is that there is a distinct Hilar mutational landscape compared to intrahepatic disease. We've seen mutations at high prevalence in mTOR and Notch1, ABL1, as a few examples, which are much higher than what's gone before. And this has significant implications for new and existing trial eligibility. The exome sequencing series has given us access to a new mutation not traditionally associated with the cholangiocarcinoma setting. It's highly prevalent at both the somatic and germline frequency, and thus has potential driver status. It is targetable, and it needs further workup, and that is work that we're going to carry on in Liverpool. Pathway analysis has shown us that there are multiple enriched immune response genes. And overall, this shows us that most of the alterations that we've seen at an enriched level are present in the host immune response. And this has given us new avenues to explore tumor infiltrating immune cells. And we're taking that further at the moment, in addition to exploring the tumor mutational burden with our single cell sequencing studies. And that's work that we're doing at the moment with single cell RNA-seq. And that is shedding new light on T and B cell infiltration and how this relates to the tumor microenvironment overall. So thank you very much for listening, and hopefully we will be back again next year to talk you through the results of our single cell RNA sequencing analyses. Thank you very much indeed.